All right, next up we have Grace Neville. She is a graduate of Smith College and currently a medical student at Dartmouth at the Geisel School of Medicine. She completed her undergraduate degree in biochemistry, and then at Dartmouth, she has been working, researching in post-traumatic brain injury at the VA Medical Center nearby, as well as studying parathyroid hormone levels after parathyroidectomy. She has a wide range of interests, but is most interested in vascular neurosurgery and neuro-oncology. She'll be presenting today on some of her research in post-traumatic brain injury.
Thank you, Dr. Ferguson. I'm going to go ahead and share my screen. All right. Good morning, everyone. Thank you for giving me this time to present. As Dr. Morgenstern said, my name is Grace and I'm coming from Dartmouth. Today, I'm going to be presenting on an animal model of a blast TBI. I joined this lab out of my interest in neurotrauma, and I wanted to gain some experience working with mice. And I had the unique opportunity to do this at a local VA medical center. So here's a table of contents of my presentation. I'm first going to talk about the neurocircuitry involved in both TBI and PTSD. Then I'll go into the goals and methods and results of our lab and talk about some ongoing work and the conclusions and limitations we found so far. All the information presenting here today is currently being written up in a manuscript for a preclinical review article that informs our model. So to start, TBI unfortunately affects up to 68 million individuals per year. Combat military personnel are at an elevated risk, specifically for shockwave-induced TBI out of their close proximity to improvised explosive devices. And interestingly, up to 80% of the mild concussive injuries seen in Afghanistan and Iran were shockwave-induced. These combat veterans are 3 times more likely to develop PTSD than non-combat veterans. And there was an interesting study in active duty Marines that was looking at predictive factors for PTSD post-deployment, and TBI was the strongest risk factor, even after accounting for pre-deployment symptoms and combat intensity. And this may explain why combat veterans are 68% more likely to attempt suicide post-deployment. TBI can lead to neuropsychiatric complications that significantly impact the quality of life and health outcomes, not only of vets, but also their families. And this creates a profound societal and personal health burden. Specifically, blast TBI has been shown to cause reduced executive functioning, spatial learning deficits, as well as the symptoms of hyperarousal, intrusions, avoidance, and reactivity that we know are associated with PTSD. Various neuroimaging studies have been shown to implicate some brain regions that I've shown here on the right, most notably the prefrontal cortex, nucleus accumbens, hippocampus, and amygdala. And this has gained the interest of various specialties, most notably neurology, psychiatry, and functional neurosurgery. And this is important because we know that a multidisciplinary approach is needed to treat neurotrauma, both in the acute and chronic phases. One such circuit that I want to highlight is the prefrontal amygdalar circuit. Biologic studies have shown that mild stress activates neurons within the amygdala, and this causes a wide cascade of activation of glutaminergic, dopaminergic, and serotonergic neurons throughout the brainstem and basal ganglia. This activates glutaminergic neurons in the prefrontal cortex, and as the body is able to adapt to the stress, GABAergic inhibitory interneurons in the prefrontal cortex ultimately feeds back and downregulates the amygdalar stress response. However, with extreme stress, the amygdala is activated to a higher degree, causing increased monoamine levels in the prefrontal cortex. This inhibits its ability to feedback and downregulate the amygdalar stress circuit. So how can we modulate this pathway? Neuromodulation, such as DBS and VNS, are FDA-approved therapies for extrapyramidal diseases, such as Parkinson's disease, essential tremor, and dystonia. Specifically, high-frequency DBS is hypothesized to activate local inhibitory interneurons, resulting in altered firing rates and neurotransmitter release to alter neurometabolism and synaptic plasticity at both the cellular and the molecular level. So in theory, direct stimulation of the amygdala via DBS should result in a reduced stress response. So animal models of PTSD they have the potential to elucidate the mechanisms behind the behavioral improvements that we see following brain stimulation. Before my time in the lab, they previously showed that phasic stimulation of the nucleus accumbens enhances spatial learning following TBI. And as you can see here in figure A, we're looking at escape latency, which is inversely correlated to spatial learning. And you can see in the blue line here that following DBS treatment, escape latency decreases or spatial learning is enhanced. And if you go from figure A to figure B, you can see that after 5 days of treatment, treated animals begin to approach escape latency of that of uninjured animals. And this effect persists after 10 days. So if you go from figure A to figure C, you see after 10 days of rest, they still exhibit an escape latency similar to uninjured animals. The lab also found that synapsin-1 and GAP43, which are markers of synaptic vesicles and neural growth cones, are upregulated following phasic stimulation, suggesting that DBS may work at the genetic level to alter neuronal differentiation and cell signaling. So neuromodulation has the potential to enhance functional recovery following TBI, but are these effects still seen after repetitive TBI, or are repetitive injuries, the effects, are they additive? So to our knowledge, we're the first lab that's looking at deficits in neuromodulation after repetitive blast TBI. You can see some of our overarching aims here in my specific aims during my summer. I was focused on learning the TBI protocol as well as troubleshooting a behavioral assay that I'll speak about. I gained some skills doing microsurgery and handling the mice both in their pre and post-injury phase. And then I also am still working on the literature review and the manuscript writing for this preclinical review article. So to get onto our methods, this is a schematic of our blast tube, and this device is similar to that used in other high-profile military research institutes, such as the Walter Reed and the Naval Military Research Center. What you see here is compressed air is going to fill the driver section and burst a mylar membrane, sending a shock wave down the tube. We have 2 pressure sensors along the tube. Anesthetized animals are placed in a prone and head forward position relative to the shock wave. Forward here. This is another schematic or picture of our blast tube, and this is a picture of what the burst mylar membrane looks like following an injury. So in order to assess the spatial learning deficits following TBI, we allow the animals to recover for 24 hours and then test them on the Morse water maze, which is a well-known assay that's been employed since the 1980s. The overall aim is to repetitively put these animals in a stressful situation and see if they can escape and learn the escape pattern. We have a pool that's divided into 4 quadrants, northeast, south, and west, and there is an escape platform that is always located in the southwest quadrant. The animals are tested 4 times per day, and they are randomly assigned to a starting quadrant and allowed to swim for 2 minutes. And Noldist ethovision is the tracker that is going to track their swimming patterns throughout those 2 minutes. And we see that there are 2 general patterns that start to appear. Either the mouse learns and utilizes a path to the southwest quadrant, and we measure their escape latency or their time to platform, or the mouse does not learn the location of the platform. Sometimes they'll just swim aimlessly for the 2 minutes, or sometimes they'll even freeze in the water. So to assess the feasibility mortality rates in the spatial learning deficits following repetitive blast TBI, I conducted the pilot study during my summer, looking at different blast pressures and interblast intervals, which is the IBI. We chose blast pressures that were previously found to induce axonal injury, chronic motor deficits, and increased pro-inflammatory cytokines. For reference, 27 kilopascals is the current safety standard for military settings, and it correlates to the pressure at which the tympanic membrane will rupture. In addition, we chose extremes of interblast intervals, less than 5 minutes being the shortest amount of time we could get 3 consecutive injuries in for one animal, and then 24 hours IBI. So what we did is we took all these groups and then tested them on the Morse water maze. And as hypothesized, we found that repeated blast TBI did exhibit an increased escape latency or spatial learning deficits significantly beginning at 5 days post-injury. And another supplementary project that I'm still working on is a review article that's summarizing the role of animal models in elucidating the mechanisms behind neuromodulatory therapies and the neurocircuitry of PTSD. I conducted a PubMed search using search phrases such as neuromodulation and PTSD and DBS and preclinical animal model, and it came up with about 7,500 articles, and we've narrowed it down according to impact factor, similar author groups, and most recent articles. And this is just a schematic of what the focus of the article is going to be. I'm going to touch on the neurocircuitry of PTSD, similar to how I did at the beginning of this presentation. I'll talk about the current uses of focal brain stimulation and some of the models of PTSD that are used preclinically, such as the PredX and the SPS model, and some of the overall limitations of animal models in general. And then finally, I'll talk about how the efficacy of brain stimulation is being assayed in animal models. There are specific tests looking at things like anhedonia, fear extinction, and anxiety. Okay. So to conclude, I have talked about how experimental brain TBI can be feasibly performed with animals, and our mortality was less than 10%, mainly in the first 24 hours post-injury. And after functional recovery, I've shown that the Morris Water maze can be used to assess spatial learning deficits following injury, and they do display significant deficits compared to uninjured animals. Specifically during my time, I gained some microsurgical techniques, and I was really excited to work with mice for the first time. However, our study is not without its limitations. We decided to solely utilize male mice in order to correlate with the male predominance in combat veterans. However, previous research has shown that there may be sex-specific differences following TBI. In addition, we're testing these animals 24 hours post-injury, and this time frame does not correlate with when veterans will present to their local VA hospitals. The temporal relationship of when there is a pro-inflammatory cytokine surge following TBI hasn't been elucidated yet, so we may need to alter the timing of our behavioral assays. Again, this work is all ongoing. I'm excited to continue its progress, and we all have the ultimate goal of mitigating the symptoms associated with TBI and PTSD in order to improve the quality of life for vets and their families. So I want to take the time to acknowledge my mentors for this work, Dr. Joshua Anderson, who's since moved on to the BI in Boston, Mass, and Dr. Crystal Noller, as well as other physician scientists at the VA in White River Junction, Vermont, and Dartmouth Health Medical Center. And again, I want to thank everyone at Science so far. It's been an incredible experience. Everyone has been so welcoming and eager to involve us sub-Is in surgical cases and patient care, and it makes me really excited for the future. Thank you again, and I'd be happy to take any questions.
Trevor, I saw your hand up first.
Oh, sorry.
Or Dr. Chowdhury. Then.
Grace, enjoyed your presentation. Very important subject. I like the fact you guys are doing repetitive injury. You correctly pointed out the limitations of doing it so acute when most of these combat people get later on. I guess the question is, for severe injury, and you had 10% mortality, I understand that that's one type of injury, but there's other people, obviously, that have more subtle injury, and those are the ones that are often repetitive. And with animals, you can't really tell the subtle symptoms. How do you think we can address that? Because I do think you're onto something. We need models, even though animal models are imperfect. So can you comment a little bit about that for the milder symptoms that you can't pick up from an animal of that size?
Yeah, sure. So I talked a little bit about the Morris Water maze. However, our lab is also looking at a array of neurobehavioral assays. So we're also looking at motor deficits on a rotor rod, which is a rod looking at, it's basically a spinning wheel to see if there's any motor deficits. There's also, we have an anxiety protocol looking at an open maze and seeing if the animals prefer to be in a secluded part of the maze or over a height. And so we're really trying to look at a vast array of phenotypes that may exhibit post-injury.
Great, thank
you. Hey, Grace, very nice work. Really enjoyed the talk. I'm kind of going after Dr. Chowdhury's question. The tests that you've mentioned thus far, Morris Water maze and open field, those are all dependent on locomotor, preservation for accurate assessment in the mice. So I noticed in your review that you were putting together that you were also looking at things like fear conditioned learning. So that's another great test for memory and anxiety. I wanted to know if you guys had thought about any other tests besides that to try and look at addressing that locomotor issue. And do you see any locomotor issues post injury in your model?
We haven't specifically like identified the locomotor issue. We've just seen the spatial learning on the Morris water maze. But there is a test also in the lab looking at anhedonia, so sucrose preference. So testing if they will take in either like sugar water or just plain water at different rates.
Thanks.
Dan, you have one too?
Hey, Grace. So I don't know if you're aware of the work Dr. Langevin at in Los Angeles. They're doing DBS of the amygdala for OCD. There was a paper in Nature Communications earlier this year on that. So they're doing it in humans, but it just kind of brings up a question that Dr. Chaudhry brought up to some degree, which is that like what things are mice models good for, especially when it comes to human circuitry. How well do you think mouse models would recapitulate the neural circuitry of PTSD in humans? And how would we validate that mice are a good model for that?
Right. So I think that's a great question, really the crux to what we're trying to get at with this review article. I think that there are things that we can study in mice, and we can, test different behavioral assays over a longer period of time and get an increased N. But I think that there is a limitation into the amount that we can do. So the neuroimaging only goes so far, and how that translates to humans. And so I think that there's a big limitation there. But I think that it's important to, we can get some brain slices and look at some cell signaling pathways through some staining that we wouldn't otherwise be able to do in humans that I think can elucidate some of the pathways there.
How do you induce OCD in a mouse, or sorry, PTSD in a mouse? How do you?
Yeah. So there are two models that I was looking at. So there's something called the PredX, or Predator Without Escape model. And that is you're looking at you're basically exposing an animal to a predator without an escape route and measuring startle response over time, see if they become capitulated to it. There's also a single prolonged stress model, and that's measuring after multiple stress response, the level of HPA activation following that.
All right, thank you, Grace, and everyone for your questions.